So once again, good evening, dear colleagues. I'm so glad that you decided to attend my presentation tonight. Please allow me to present our research on interaction of broad host range bacteriophage VBPMITS with bacteria of Proteus genus. Proteus genus bacteria are quite notorious for causing a vast array of diseases as well as their antibiotic resistance. In clinical practice, most common Proteus infections are urinary tract infections or UTIs for short. In nosocomial or hospital-acquired UTIs, Proteus accounts for about 5% of all cases. Complicated UTIs, such as catheter infections, have an even higher association with Proteus bacteria. Thus, development of novel therapeutic methods is of great importance. One such method is the use of phages and their derivatives. Broad host range, BHR for short, phages are especially promising. Hence, BHR phage discovery and detailed description of their interactions with host cells is very much crucial for modern phage therapy advancement. The aim of the present study was to isolate and characterize the Proteus-infecting bacteriophages from plant material, which was infected with microbial association containing Proteus genus bacteria. From a crude lysate obtained from homogenized tomato specimens, a pure line of bacteriophage was obtained after a 5-fold passaging through a single negative colony. The obtained plaques were 1 to 2 millimeters in diameter, although some of them featured a halo of lysis. Such heterogeny was observed even after further passaging of plaques. In order to study phage morphology, transvision electron microscopy of singular plate was performed. Dissected phage particles featured B1 morphology, typical for Cyphoviridae family phages, with an icosahedral capsid with maximal diameter of 50 nanometers and a long, non-contractile tail. Some phage particles or individual tails, as you can see, were attached to the flagella, which may indicate the probable flagellotropic nature of the phage. Another interesting finding were the lysed host cells, all at different stages of lysis. From this point on, further studies involved characterization of the phage host range. Beforehand, the host bacterium species assignment was proven. After 24 hours of cultivation, host bacterium demonstrated classic Proteus mirabilis colony morphology with concentric circles on the agar plate. Cell morphology also matches Proteus mirabilis description in literature. Biochemical profiling is consistent with reference values in Burgess manual as well. Reactions clearly demonstrate the difference in compound utilization between Proteus mirabilis and Proteus vulgaris. 16S RNA gene fragment sequencing also proved the host bacterium to be Proteus mirabilis, as its obtained 708 nucleotide sequence featured 100% identity. Let me remove the bar so you can see the 100% identity with that of representative genome of Proteus mirabilis HI4320 at GenBank and CBI. Along with the phage host, 10 strains of 3 species of Proteus genus, among them 5 were clinical isolates, as well as enterobacteria, E. coli, C600, and Erwinia species were used. The studied phage was able to infect all 3 species of Proteus genus, Proteus mirabilis strains ATCC 8504 and DSM 6674, Proteus vulgaris B905T, and Proteus houseri DSM 3118. But it should be noted that none of the clinical isolates were susceptible to the phage. Enterobacterial cells also remained resistant. Thus, though featuring a broad host range capability in itself, the phage cannot be recommended for use in phage therapy, though it still represents an interesting object to investigate the mechanism of attachment to 3 different bacteria species and causing a productive infection within them. Apart from that, we have also noted the presence of polysheaths in obtained micrographs, and their production may explain the sporadic occurrence of lysis halo around phage plates. Since the studied phage is a cyphophage, and hence does not contain a tail sheath protein in itself in its virion, our hypothesis was that an induction of defect prophage elements has occurred. As the genome of host bacterium is not sequenced, we decided to check whether such tail sheath protein encoding genes are actually present in available sequenced genomes of Proteus bacteria. Thus, we have studied the occurrence of prophage elements in all fully sequenced genomes of Proteus vulgaris, that's 11 sequences, and 20 sequenced Proteus mirabilis genomes available at GenBank and CBI. All 31 genomes were shown to contain multiple intact or incomplete prophage elements, from 3 up to 14 per genome. In order to specifically search for tail sheath coding genes, we have performed manual annotation of all genes comprising the prophage elements of 3 Proteus genomes: Proteus mirabilis HI4320, Proteus vulgaris FDA August 556 and 1507. Among the identified genes, we have determined 6 tail sheath proteins that, on basis of sequence similarity, were assigned to 3 clusters. After that, we have performed amino acid sequence comparison of the cluster representatives against the whole proteome, as you can see, of all selected Proteus vulgaris and Proteus mirabilis genomes. As a result, we have identified the presence of tail sheath protein coding genes in each analyzed Proteus mirabilis genome, while in Proteus vulgaris, 2 genomes out of 11 did not contain any. From the obtained results, we have constructed a phylogenetic tree. Here you can see 4 groups colored in purple, with Proteus mirabilis gene fragments clustered nicely together and demonstrating similarities with reference lysogenic phages. A smaller blue group, it was more diverse, with both Proteus vulgaris and Proteus mirabilis specimens together, sharing similarities with P2 temperate phage. A green group comprised of Proteus vulgaris fragments and similar to, once again, lysogenic phages, and a pretty remote group colored in orange, clustered with reference lytic phages. Again, in all but 2 analyzed genomes, there were tail sheath protein coding genes. Thus, we can expect that our strain also contains such genes, and the structures observed under the microscope can thus be explained. So, to summarize, a broad host range cyclophage able to infect collection strains of 3 species of Proteus genus was isolated and characterized. The phage was proven unable to cause infection in clinical Proteus mirabilis isolates, and thus cannot be recommended for use in phage therapy. Following the detection of polysheath dibacterial sins, and based on publicly available data, diverse phylogenetic groups of tail sheath proteins were predicted, laying ground for further research. Full genome sequencing may also help shed light on multiple host infection mechanisms. And that concludes my presentation. I would like to take a moment to thank my incredibly caring and supportive supervisor, Yulia Paiduk, for all her help and feedback, as well as Alla Harina and Pavlina Zelena for their irreplaceable input on this project. And that's all from me. Thank you.
Thank you so much, Daria. Really very interesting topic to represent to us. So, do you have any questions, dear participants, dear colleagues?
Якщо можна, коротко тоді спитаю. Ви виділяли з рослин ваші фаги, наскільки я розумію. Які їх подальше практичне застосування ви пропонуєте?
Саме у цього фага, який ми виділили, правильно?
Так, так.
Саме у цього фага практично в медицині, на жаль, ні. А от якщо ми зможемо направити його в якусь систему, наприклад, взяти ряд бактерій і просто досліджувати, як цьому фагу вдається інфікувати різні бактерії філогенетично віддалені одне від одного, якщо ми знайдемо цей механізм, яким він користується для інфекції багатьох бактерій, то це було б дуже круто.
Ясно, дякую, зрозуміло. Дякую.
So, dear colleagues, I guess this is it. We finished already up to during our timeline.